So prostaglandins and thromboxane are two other major eicosanoids in addition to the leukotrienes. Their synthesis is also very interesting and important topic in as far as the metabolism of lipids is concerned. As was the case in leukotrienes, in the case of prostaglandins and thromboxane, for the sake of simplicity or convenience, we will say prostaglandins as PG and thromboxane as TX. So both PG and TX are also their synthesis is initiated by the arachidonic acid. The arachidonic acid is the main precursor of these compounds. Arachidonic acid is a omega-6 fatty acid with 20 carbon atoms and 4 double bonds. The omega comes from or omega-6 come from the double bond, the presence of the double bond on the presence of the double bond on the carbon atom number 6 in these compounds. It is derived from the linolenic acid, another very important fatty acid. It is incorporated into the cell membrane, in the phospholipids of the cell membrane, typically the phosphatidylinositol. There are two series of PGs. One is that is derived from omega-6 eicosanoids, and the other is the series 3, which is derived from the omega-3. As far as the synthesis pathways are concerned, the main enzyme which is involved is the cyclogenosis. There are different isozymes of the cyclogenosis depending upon the tissues in which these compounds are being formed. First of all, if you see here what we say, the arachidonic acid acted upon by cyclogenase along with molecular oxygen, and you get the prostaglandin G2, PGG2, which is then converted into PGH2. Then glutathione is also used as a supporting enzyme for this or supporting substance for this. COX-2 and COX-1, these are the two enzymes which are specifically or mainly involved in the pathways which leads to the production of different prostaglandins and also thromboxane. COX-2 actually is it occurs in almost or most of the tissues, while COX-1 occurs in most of the tissues, while COX-2 it is produced mainly in the immune system cells. So what happened that once you get the PGH2, which I have already told you how it is produced, then it can give rise to the formation of TXs and different PGs, that is different prostaglandins and different thromboxanes. And because they are being produced in different tissues, so it again depends upon which enzymes are, which enzymes are making these compounds. It depends upon in which tissues this process is taking place. And of course, then accordingly, the functions these compounds produces also depends upon the tissues in which they are being produced. For the sake of I am not going in big detail of this, but just for the sake of your interest, let me tell you that, for example, PGI2 is also one of the compound which is formed by the PGH2. It is acted upon by the it is formed because of the action of the COX-2, and it is involved in a very important process, the vasodilation, which leads to the relaxation of your arteries. Similarly, PGF20, another prostaglandin, it is also involved in the vasodilation and also vasoconstriction and the bronchoconstriction. And also then your urination process also depends upon the activation of the contractile system, which involves into the urination process, and it is stimulated also by the PGF20. So in this way, you can see, depending upon the tissues, the type of prostaglandins or thromboxane also changes, and also you can see how diverse are these compounds.